Now we will discuss about the third experiment which convinced that DNA had uh, a variety of chemical structures and possibility of allowing biological specificities by Erwin Chargaff, the chemical experiment. The Chargaff rule, it is also called as Chargaff's rule. Chargaff applied new separation and analytical techniques that showed that the molar concentration of bases could vary widely. Thus, it was demonstrated that DNA could have variable compositions, a primary requirement for a genetic substance. That is a primary requirement of a genetic substance. The hypothesis of tetranucleotide structure for DNA arose from the belief that DNA contained equimolar quantities of adenine, thymine, guanine, and cytosine. This incorrect conclusion arose for two reasons. First is first in the chemical analysis of DNA, the technique used to separate the bases before identification did not resolve them very well. So the quantitative analysis of so the quantitative analysis was poor. The second reason for incorrect conclusion was that the DNA analyzed was usually isolated from eukaryotes, and in eukaryotes, the four bases are nearly equimolar. And from bacteria, those DNA happen to have already nearly equal amounts. Using the DNA of a wide variety of organisms, Chargaff applied new separation rules or separation technique and analytical techniques that show that molecular concentration or molar concentration of bases could vary widely. Thus, it was demonstrated that DNA could have variable composition, the primary requirement of a genetic substance. Upon publication of Chargaff's result, the tetranucleotide hypothesis quietly died and the DNA gene idea began to catch on. Shortly afterwards, workers in several laboratories found that for a wide variety of organisms, somatic cells have twice the DNA content of germ cells. The characteristic to be expected of the genetic material. This given, given the tenets of classical chromosome genetics. although, it, although it could apply just as well to any component of chromosome, once these results were revealed, the objections on the work of Avery McCarthy were no longer heard, and the hereditary nature of DNA rapidly became an fashionable idea. The Chargaff rule contained one other key piece of information related to the structure of DNA came from the Australian biochemist Erwin Chargaff. Chargaff analyzed the DNA of different species, determining its composition as A, T, G, and Z, that is adenine, thymine, cytosine, and guanine bases. He made several key observations, that is A, T, C, and G, that is adenine, thymine, cytosine, and guanine, were not found in equal quantities as some models of at that time would have predicted. The amount of the bases varied among species, but not between individuals at the same same species. The amount of adenine always equal the amount of thymine. A always equal T, and the amount of cytosine always equal the amount of guanine. That is G always equal to C, or C always equal to G. These findings, called Chargaff rules, turned out to be crucial to demonstrate the Watson and Crick model for DNA double helix, and it was the final blow for the hereditary nature of DNA. And the explanations that are hindering the Avery McCarthy experiment were being eliminated by this Chargaff rule. So we'll discuss in next point the blender experiment.